Welcome to the Earthworks podcast, where our team will share the jargon of carbon from many of our turf friends from the past 30 years.
Hi, everybody. This is Joel Simmons, and this is the Earthworks podcast. I'm very excited. We have Laura Decker with us today, who is the president of MicroBiometer. And this is a tool, and I think, Laura, I told you this, we own 2 of them. We'll own 4 by the end of the spring, maybe more, depending on how many agronomists we are. But those folks that have listened to me over the years know that our passion of all things that we do is soil biology. And the hardest thing in the world is to quantify this. And when we first read of you folks, we were very excited because you've brought to the table a handheld meter that we can use in the field. And I'm going to have you talk all about it and kind of give us some insight on that. But it's like I've said many times in some of my presentations. I hope by the time I'm done on this earth that we have a better understanding as to what biology is really doing. Our chemist, and I think you know of Lawrence Mayhew. He insists that we have cataloged 5% of the soil biome. Is that what you, I mean, actually, I say 5%. He actually says it's more like 2%.
Yeah, I was going to say 5% is probably too high.
Yeah, he tells me the same thing. I tend to exaggerate a little bit as I go through.
Yeah, and the thing that you need to know is soil biology mutates incredibly quickly.
Yeah.
So it's not like if you learned it one day, you'd be done, because they, with such little DNA, their evolution is like viruses, is quite quickly. So the population does change quite rapidly.
Yeah.
So if you know it today, you might not know it in a couple weeks.
And just to be fair to our audience, much of your work is really in agriculture.
Yes.
You're relatively novice in the golf course world, I assume. Is that fair? Or are you doing some work?
We sort of go wherever people will take us.
Good. Well, we're taking you there. So you're here now. And we want you here.
We would like to be in golf courses. We have tried to reach those markets. I'll give you a little intro of our company, but if any of you work at small companies or startups, you'll know that you do the spaghetti on the wall, right? You said, right. You're like, this week I'm going to see if golf courses will be interested, and you send a bunch of LinkedIn requests and emails, and if people write you back, then you're in. If not, you move on to cannabis growers or whoever's there.
Where the business is.
Yeah. So,
it's funny because in our world, and I shared this with you before we started. I mean, the golf course, the athletic field, even a lot of the lawn care clients that we work with, but certainly the professional side, these are forced environments. I mean, the golf course, Mother Nature didn't create the golf course or the soccer field. And many of our properties are sand-based. So they build the greens, the soccer fields out of sand, and inherently that eliminates a big portion of the soil biome. And it's a struggle. And Earthworks has been doing work for 37 years now, trying to basically feed microbes, but we've never really had a really good way to quantify and qualify that. So it's been criticized. And, one of the things that I say all the time, and I think you would support this, is that biology is not a reductionist science like chemistry can be. It's a dynamic science. So it's always expanding. And like you had just said, it's changing. How long does a bacteria live in the soil? 20 minutes?
Yeah. I mean, and they reproduce,
constantly.
Yeah. They're quite, as my mother would say, promiscuous.
Yes, they are. Rabbits have nothing on microbes. Yeah.
They live fast and die hard, right?
Yes, they do.
So yeah, it's terrifically exciting. Why don't I do a little introduction to myself?
Yes, please do, and tell us about how you guys got started.
If they want to believe anything I say, like you said, my name is Laura Decker. I'm the president of MicroBiometer. We are a very small 4-people company. We were located in New York State, but we just moved to where I live, Washington State, which is terribly exciting for us. We developed, manufacture, sell, and market the MicroBiometer. It was developed by Dr. Judith Fitzpatrick, who happens to be my mother. It's always fun to work in agriculture because everyone thinks a family business is quite normal. So
Right.
it's quite fun. She had a passion for microbes. She studied human immunology for a very long time. Microbes do the same thing, whether they're in your gut or in the soil, right?
Yeah.
They're symbiotic beings, and they live a very social life. Social meaning lots of them. And she wanted to develop a test, particularly. She knew the importance of soil biology. She knew that people were not understanding how important it was, and that they were only focusing on chemical. So sort of the chemical factors of life. So you have a plant, it likes nitrogen, you feed it liquid nitrogen. It's essentially, she always likened it to just putting an IV on a person and feeding them, sugar, and then wondering why they're not healthy, right?
Yeah, hydroponics.
Yeah, hydroponics. So we developed the microbiometer. People who are listening won't be able to see it, but it looks exactly like a little lunchbox. It's plastic.
It is a little lunchbox.
It is a little lunchbox.
Yeah.
She believed, and we believe, soil is a living thing. If you take it out of the soil and dry it on your dashboard and ship it in the mail for a couple weeks and it sits on a counter, when you test it, it will be different than when you took it out.
Yeah.
So she wanted a test that was, you go in, you take the soil out of wherever you want to test, as close to the roots as you can, and you test it right away and you get a number. And then you use that number to assess what's going on, to have visibility into the biology of your soil. So what you do is you go into the soil, you grab a little soil sample, you take it back. We have an app with instructions, videos. We're in 19 languages. So if you speak another language, you don't have to worry about English. If you're trying to learn another language, perfect. Use this
There
as
you go.
practice.
Trying to learn Italian right now.
Yeah, do Italian.
Yeah.
You do the little test. It will make you feel like you're in high school again. You mix stuff up and you let it settle, and then you use your cell phone. The other thing we wanted is there are very expensive machines and satellites and things that you can use, but they're not accessible to a lot of people, right? And so she wanted it to be really accessible to people. What is available to a lot of people are smartphones, okay?
Yeah.
So we use a smartphone app camera to colorimetrically read this microbial soup. Now, microbes, particularly in very, very healthy soil, are tightly bound with soil particles. They make soil structure by sticking to clumps of soil, right?
Yeah.
One of the things, you can't just take a blob of dirt and put it under a microscope, right, and see stuff. You have to do something. So most people put it in water and shake it up a little bit. Well, we added a bunch of research. It took about a year and a half to figure out if you put some salt solution into it and you whisk it in a controlled way, you will mechanically and chemically work to separate the microbes from the soil particles. And then we use very expensive technology called gravity to allow the soil particles to precipitate out. And we get basically a microbial soup. Now, you have to know objectively that some of the microbes are killed in this process. Ultimately, all of them are, but and some of them do precipitate out. So it's very important that the timing of it is consistent, right?
Are you only testing live microbes?
Live and dormant microbes.
You are, okay. So the ones that we have killed in this process,
we thank them for their service.
Right. But there's billions of them, as Dr.
There's
Sagan
billions
would
of
say.
them, yeah. So in this microbial soup, we let it settle for 20 minutes, which is sort of the sweet spot of where we tested all the soil. And if you do it every time, then your soup will be the same sort of die-off. You put the stuff. This only works in natural, real soil. So not hydroponics, not soilless medium, because we rely on the natural pigmentation that microbes, fungi, and bacteria pick up in the soil, okay?
What about sandy soils?
It works for sandy soil. The only thing we'll say about sandy soil is if it's very, very sandy, the sand will precipitate out quite quickly.
Yeah.
So if you can control it, you can change the settling time to 5 or 10 minutes. It's very important that if you're going to change the settling time, that you do it uniformly. Because if some samples are tested at 5 and some at 20, the one at 20 will naturally be lower because of die-off and precipitation.
That makes sense.
So you want to, it's a scientific method. All of us are going back to high school. You want to control your variables and keep things as consistent as possible. So if you have very, very sandy soil or very, very heavy clay soil, you might want to reduce the settling time. We are working on getting some guidance in the app to have people understand their soil consistency and make those decisions. But that
Good.
is we're going to be called an advanced feature, so that if you have no idea what the word soil composition or soil texture is, then you shouldn't be doing it. But if you do know, then you can make those decisions. So you put it on the test card, and it returns back micrograms of microbial carbon. Microbes are between 50 and 60% carbon, so we just return it as microbial carbon and fungal to bacteria ratio. What people do with this information is they try to figure out what's going on in their soil, and more importantly, what is the effect of their practices on their soil biology, right?
Yeah.
So they are using different amendments, different biologics, different input. Agriculture is sort of where we work, right? Because that's most of the soil work that's being done. Turf is a crop, right? I mean, one of the biggest crops in the biggest crops in the United States is lawn. Okay, we grow lawns all over the place. That is also essentially a crop. All crops are artificial, right? In the natural world, you don't have mono cropping. You have lots of stuff, and a lot of cool people in agriculture are returning to that and doing intercropping, multi-cropping, and all kinds of stuff to get the biological diversity up.
Yeah, unfortunately,
so
we won't ever have that option.
No.
We won't have rotational grazing and intercropping. I mean, we might have different varieties of a grass plant.
I mean, even different varieties of grass is something, right?
Yeah.
But I know what my husband has a degree in turf management, so.
Oh, I didn't know that.
Yes. He works in horticulture, which is soilless. So, which I find very sad, but, and horticulture is just a chemical.
Yeah.
That we won't talk about, but
yeah, let's not go there.
Let's not go there. So we work primarily in what we call sort of regenerative or climate friendly, or all those nice terms, and organic, of course. Yeah, sustainable. All those words work. It just means people are out there trying to figure out a better way to grow, which is what farmers have been doing since we just not discovered farming, but when we started cultivating crops. So the test is used. So if people are trying different practices, they test before and after. They get a sense for what's the natural variation in microbes throughout the growing cycle due to weather, and also what plants are doing when they start growing, when they start to reproduce. They behave with biology differently. One of the most important things for people to understand is that plants and microbes are symbiotic. So what a plant does, we were just talking about this for another presentation. What a plant does is put leaves into the atmosphere, grab carbon, right? Put it into the roots and barter with microbes for the carbon. Microbes need carbon. They can't get it themselves.
I like that phrase, barter. I think I'm going to steal that from you.
You should. I stole it from someone.
I'm sure. Yeah, it's good.
They barter. They say, I have this substance. Let's trade. And they do trade. If people want to go super into the wormhole, rhizophagy is one of the coolest things. And then that carbon that the plant took from the atmosphere goes into the microbe in the form of microbial carbon. And then that microbe dies, right? That microbe will die and become soil carbon, right? Which hopefully humans, and then that carbon will either get eaten by another microbe or it will stay as soil carbon. And then when plants aren't growing and aren't, in the winter, right? They're not doing as much activity. The microbes will stay alive by eating that stable and humus carbon until the plants start bartering them again. We talked about biological or genetics of microbes before. One thing to remember is that a microbe only has a couple strands of DNA, and they only make one or two substances. So what happens, for anyone who's ever done it like a petri dish thing, the petri dish will start, say you have a little gross petri dish and put your hand on it. You want to prove to your kids that they should wash their hands. The first thing you'll see is one color, right? Yeah. And then in a couple of days, you'll see another color. And then in a couple of days, another type. right? And what's happening is that microbe A starts to grow and it makes substance one. And then microbe B will start to grow and it'll make substance two. And A and B will trade with each other, right? Because they both
Biotic.
need those
Yeah.
substances. And then C will come up and make substance three. So microbes need a lot of things, but they can only make certain things. It's like being a cobbler, right? I can make shoes, but I need bread, right?
Right.
It's if you picture a medieval village, that's exactly what it is.
Oh, I
Everyone
like that too. I'll
specializes
steal that from you as well.
and they barter. Yeah, I keep thinking of these video games our kids play where they make
Yeah.
little villages and they grow, and that's what microbes do. And the plant is sort of the ultimate carbon giver. And as we talk about carbon sequestration, and there's no reason why golf courses can't rehab their reputation as sources of carbon sinks and
They're
get
cover
into the
crops,
carbon
so they
market.
are. I mean, they're one of the best, to be
Yeah.
honest with you.
I mean, at some point, carbon markets are going to be, a real thing, and
Yeah.
you can make money on golf, and you can,
Sell carbon.
have a seal that says we have a net negative carbon, and we have so much carbon that we sell it to other companies.
Yeah.
So that's how the carbon kind of gets into the soil. I mean, there's more complex. It's a very complex world. But when you do want to grow soil health, you want humus, which is carbon matter in there to sort of take the microbes through famine, right? It's sort of like microbial food stability.
Survivability, yeah.
Yeah. And then the other thing is the more plants barter with microbes, the more roots they need, right? Because they want to reach more microbes in different places
Good
to
point.
do more trading.
Yep.
And the more roots you have in the soil, the more stable your soil is, the more organic matter you have in it, the better aerated it is, because as the roots die off, they leave air pockets for both water and oxygen that microbes need. And you get, instead of hard packed soil, right? You get this rich, crumbly, good stuff.
And it's easier for the plants to get nutrients because there's more roots.
Yeah. So we talk about vicious and virtuous cycles, right?
Yeah.
When you start paying attention to soil biology, feeding soil biology with your inputs, you start a virtuous cycle, right? If you stop doing that and you start doing what we have been doing, which is chemically feeding plants NPK, and plants are like people, they're lazy, right? We all want to conserve energy. So if you give a toddler a great plate of kale or a candy bar, right, they will eat the candy bar. They will not be healthier. They'll maybe grow, but kind of, in a spindly, gross
Spindly
way.
way, right?
Yeah, the plant, not the kale.
And they won't touch the kale.
They won't touch the kale. So if you feed them liquid nitrogen, NPK, the plant will be like, microbes, I don't need you. I got this, Red Bull, and they'll suck it up. And then when we talk about food, because of course food is, you're not growing food, but when you talk about nutrient density and the quality of the food that's
But
there.
that's important. You can feel free to talk about that because that's really the backbone of regenerative agriculture.
And I will gloss over it, but I am not the person that proves this or knows everything about it. But we do know that the antioxidants come from fungi in the soil, right? Your kale and your spinach doesn't have antioxidants if it wasn't exposed to pathogens and microbes, right?
Yeah, and Laura, in the turf world, that even though we're not making a food product, still having that density of mineral in the plant is going to make the plant stronger, more resilient to wear and tear and to the stress that a turf environment would have, and it would make it stronger to fight off pathogens and even weeds.
And it will look better. It will be greener.
Better. Yeah.
It will be, because it will have more of an incentive to harvest carbon, right? Because it's working. You do know that, sickly plants are not, they're a different color than.
Yes, we should by now. Yeah.
Yeah.
What's wrong with that one? Yeah, that doesn't look good.
So the microbiometer, we sell it direct from here. It's relatively inexpensive. It's $135 to buy the kit. It has 10 in it, and then people do refills. And the point being, if you're trying a biological product or even working on understanding how pesticides and herbicides impact your soil in the long term, you can say, Joel told me that if I put down pesticides, I'm going to be harming my soil biome. Let's find out, right?
Yes.
I'm going to test before, I'm going to test after, right? You also need to know that lots of things happen in the real world. So if there's a drought or, it gets cold, that will also impact it. So you have to use your brain when you look at this data, as you do with everything else. And so people use it that way. And then our app is there. You can store the data in the app. We're adding a feature pretty soon where you can put a picture, right? So you can be like, oh, my reading was 300. This is how my soil looked. This reading is 600. How does it look? Or my plants? And you can also create groups so you can share data along with people.
Right.
And of course, those of us who don't love looking at teeny screens can go to the cloud and get it in a big format for our old eyeballs.
Is it all databaseable? Is all the data something that we can now date? Can we then track it and compare test to test to test?
Yeah, yeah. I mean, you'd want to name them so you're not comparing
Right.
totally different things. So when you give a good naming convention, you want to make sure the geolocation is correct. We geolocate when you take the samples. There's a time and
Okay.
date stamp.
And you
So
can
you sort
set
of
that
know.
yourself. So I can call
Yeah.
it green one if I wanted to, or fairway one,
Yeah, you can say,
and always
plot
test
one.
that site. Yeah.
And then you can, if you want, of course, no one does, but you can add custom fields, especially if other people are doing the testing. You could be like, put in when the last time it rained was, put in, like for you, turf, when did they mow last?
Yeah.
Or
When do they
put
spray
in,
last? Yeah.
yeah, when did they spray last? Or what you could do, because you probably have that information in some other database, is play the exciting Excel game. I feel like agriculture is like how finance was like 20 years ago, where you just took all these spreadsheets from different places and like
Yeah.
cut and pasted them together because there wasn't anything, you know, uh, that sort of did it. And now it's so cool. You just hit a button and all your information gets put together. So pretty soon we'll be there that people aren't having to, cut and paste. But
Yeah.
I kind
I
of feel
like it.
like probably John Deere has stuff, but that's not really in the
Yeah,
space we're in.
there's the technology is changing, but the biological piece of it is not so fast. But you guys have really started to break through some of this. Explain to me, I mean, and I think I'm not alone here. I am red-green colorblind, which is weird for being in a turf industry. But I assume that it's all done on this, so I don't even have to worry about the fact that I might not be able to qualify the colors as clearly as I would like to.
I think that is so funny because one of the things that my mother learned developing this was um, roughly 10% of men in
Men,
particular
yeah,
have some form of color ignorance. Let's put it
right.
that way. They
Deficiencies.
can't discriminate a lot of colors. Um, and and so we have a non-tech version because there are a number of Amish and Mennonite
Oh,
users for us.
okay.
And we didn't
Yeah.
want to exclude them because in the United States, they're quite a progressive group.
Yes,
But
absolutely.
a lot of them are colorblind. And instead of using the phone app, we have this, it's taken from anyone who does knitting. It's
Oh,
taken from
yeah.
something in knitting. My mother was very creative.
Yes, that's
She
very
want
creative. Yeah.
she wanted the test to be very cheap, so we sort of sourced things. Our whisker, which is what the complicated mechanism that we use to separate microbes, is actually a repurposed coffee frother because it's a
You
dollar
told
or
me that
two
at
and it's
Acres.
easy to get. Yeah.
That's funny.
So, but the color blindness is real.
Yeah.
And so it's not going to hamper you because the cell phone can see color quite
So I
well.
can figure.
But it is
Talk.
funny the things you learn when you're
Yeah.
discovering technology.
Talk to me. I mean, help me better understand. So is it actually identifying fungi and bacteria, or is it testing the carbon residual or the residue? I mean, what's the actual mechanism of what
Yeah.
your tool is using?
So what it does is we talked a little bit about sort of making what I call the microbial soup, and
Right.
then we have a specially designed test card that has a membrane. We think one of the best ways to learn about soil biology is through a microscope. That is abs by far one of the most terrific ways. It is also very difficult, not quite scalable, and for people who do it, shockingly tedious.
Yeah.
Um,
I've done it and it
and
is.
yeah, yeah, no one likes it.
No.
The first 10 minutes are super cool. Hiawassee, I don't know if you know them, they're a really cool company. They just developed kind of a portable microscope to take in the field, and it has everything in one, and it has photofluorescence. And whenever we're at a show with them, we take our microbial soup and we look at it under the microscope, and it's super fun. But I wouldn't want to do that for hours at a day. So you take the microbial soup, you put it on our membrane, and it sort of flattens out the liquid, right? So that what you have stuck to the membranes are microbes, bacteria and fungi. And then we talked about natural soil. Many, not every single one of them. There's always some scientist who freaks out at me. What about
Right.
this kind? They're clear. And I was like, okay, we're not counting those, but that's okay. There's
It's all
billions of them.
relative. Yeah.
Yeah.
We're getting a good start here.
Yeah. We read them colorimetrically. So it is designed to sort of mimic microscopy, which is how digital microscopy is how we developed it and made sure it worked. As we were developing the test and doing all this data analysis, the two founders discovered that the fungi pick up colors slightly differently. So the fungal to bacteria ratio is sort of an approximation of those because they do appear to the app differently.
Yeah.
Of course, this is a quick and easy test. Microscopy is better. It differs in method and what it's testing from respiration. People always talk about respiration. Respiration is a test of microbial activity, so release of carbon. So essentially, we respire as well. When we breathe, we release carbon. So it is a different kind of test. It is a useful test, but it's not the same as ours.
So you guys are quite advanced from what we were doing 10 years ago with respiration.
Yeah, I mean, so respiration is not used in a lot of other countries.
Oh, really? I didn't know that.
No, it's primarily something the U.S. likes. And Judy's feeling was that if you have a lot of very stressed microbes working hard, you'll have a high number. And if you have dormant microbes, you'll have a low number. And it is a useful measure of activity because it can be I mean, you do want your microbes to be active, right?
It makes sense.
Yeah, it makes sense, but you have to think nuancedly about how you're using it. Just like this test, it's not a functional test. It won't tell you, people ask us all the time if they're good or bad microbes, right? It won't tell us if they're aerobic or anaerobic. And it won't tell us if they're, we don't have, it's not a pathogenic test.
Right.
There are those tests. This is a different test. So, every time you use one of these metrics, you need to be you should be a little critical. What is it testing? What are its limits? This is a $7 test. It is not going to give you the information that chloroform fumigation extraction
exactly.
or biomakers genetic testing will give you, right?
Yeah.
But people do all kinds of tests.
Yeah.
So
talk about the genetic testing. I mean, that's DNA testing, I assume you're talking about.
Yeah.
And there's a difference there too. And my concern, and correct me if you think I'm wrong, but you could have DNA in a soil that could go back tens of thousands of years, right?
Again, this is I like how I said it before. This is a very topsoil understanding of genetic testing today. Yes. Kind of the highlights. Biomakers is one that people do the most. They do genetic testing now. There's a lot of users out there that use us and Biomakers. Biomakers is several $100s a sample. It takes a long time. And you can't really scale it up, but it is interesting information. So people put them together and say, well, I'm going to use the MicroBiometer for sort of quick and easy on test, understanding, and then I'll do deeper genetic and soil chemical testing as well. My understanding, and of course it changes over time and will get better, is that what they do essentially is we do have an understanding of the function of certain microbes. So we know about nitrogen fixing, and other key big functions, right? And those microbes that, let's for example say fix nitrogen, they look at the genetic component of those, okay, and they can identify them. And then when they get a soil sample, if there's microbes or DNA that looks like that DNA, they assume and extrapolate that it is nitrogen fixing. So they give you a functional group. You have, I've seen the reports, but I can't remember what they are. But like you have 30% or this many nitrogen fixing and this many, phosphorus fixing, whatever microbes.
Right.
and you're low in nitrogen fixing microbes, so let's do something to get more, right? To do something. It it's not that they look at the genetic code of every microbe that's in your soil sample, because first of all, we don't know them.
You couldn't,
Um,
yeah.
we couldn't, and we don't really know what all of them do. So it is a very useful generalization of the data that we have, and it will get better over time. And it will say, maybe your crop is phosphorus deficient, not because there's not enough phosphorus in the soil. So don't just throw some in, but because you don't have enough microbes that are bartering phosphorus with the
Yeah,
plant,
yeah.
right? Now that of course leads to Bayer coming up with a superbug, right? That will mine nitrogen into the soil. I don't love that idea. I think it probably is not necessary. Billions of years of evolution have
Yeah.
led us to
Microbes do that
this.
well, right? Yes, let
Yeah.
them alone. Leave them
And
alone.
and what we say is microbes literally fall from the sky. Okay.
Yeah,
So
that's
they're
a good point.
everywhere, right?
Yeah.
We learned this during COVID. There's germs,
Everywhere,
there's
yeah.
microbes everywhere. If you have soil that is dead and unsustainable for life, you putting microbes in them will not do anything. I
Right.
think we talked about this before. What you need to do is make an environment that the microbes that exist all over the place can make a good home and build their little medieval village. So
Yeah.
you want to do things like natural fertilizers. Even tilling at times is sort of necessary, even though most people tilling really destroys fungal content quite quickly,
And burns out
um,
carbon.
and burns carbon because
Yeah.
it sort of releases it.
Yeah.
But everyone knows that there are times and places where
It
tilling is
needs to
just
be done,
necessary.
of course.
Yeah, but you don't have to do it all the time, and
Yeah. I mean, in
all
our
that stuff.
world,
But
we aerify because we get so much physical compaction. So the soil just physically is so compacted because of foot traffic.
yeah,
Aerification comes in, but one of the best practices that we follow is putting carbon back into those holes to feed the microbes that we're trying to proliferate.
yeah, and so there's a way to say we're doing this aeration
Yeah,
that is somewhat damaging, but we know how to mitigate that at the same
right,
time. So, and of course, compacted soil without space for air or water is not a
right.
good home either.
No,
So, yeah, and you're growing grass, which really can have a very shallow root system.
yes, it does.
Yeah, I mean, and if it's
Relatively speaking, to crops.
yeah, relatively big.
Yeah,
And if you don't manage it well, it can have a very,
yeah,
right, unresilient root system. So, you do want to say, even though this is grass and it can only have so big a root system, we want to make it as as vibrant as we can
and
because
dense.
it'll be
Yeah,
more resilient, right?
yeah,
Like we'll have to aerate less, which is good. We'll have to put less stuff which costs money onto the turf,
yeah,
right?
yeah,
I mean, ideally, all your people don't just love grass and want to grow it for fun, right? They're making money.
yeah.
And
That's the business.
the cheaper it is to maintain that turf in a way
Yeah,
that is the product they want, the better off they'll
yeah,
be. And of course, you guys are subject to consumer pressure, which is
yeah.
I don't want to golf or be a member of a place that is contributing to watershed degradation and runoff of herbicides and pesticides. So you're having the same thing that the kale farmer is
Yeah,
facing, right? Which
yeah.
is consumers will pay more for a more environmentally thing. But I think you should start talking about carbon credits for
I like that. I think
I
there's
love that
already
idea.
some
I'm sure
conversation.
I didn't come up with this
No, it's
idea.
not. Yeah, but but again, we're a cover crop. So if you think about it, I mean, we're constantly sequestering carbon. Let me ask you this with your test.
Mhm.
How do you distinguish between bacteria and fungi within this test? I mean, is it just this?
Color.
It's all, but so
Yeah.
the fungi will put off a different
Mhm.
residue, if you will. The residuals from fungi are different than bacteria, and you can test that.
Yeah. Yeah.
Can you
Yeah.
qualify between a generic fungi and say a mycorrhizal fungi?
No,
So
we can't tell you, what kind
okay,
or what type. We do know that the people who are interested in fungal to bacteria ratios are really interested in it. It's the only thing they care about. They love fungi. They want all fungi all the time. mostly because they're building soil structure, right? They're in prone areas,
Right,
or
right,
they're growing high value crops that rely on very unique taste profiles. So
right, right.
coffee, tea,
Oh, interesting. Oh,
berries,
okay. So they want to have things pulled into that are going to be done by
grapes.
fungi.
Yeah.
Yeah, yeah, that makes
Yeah. So
sense.
when people talk about terroir, the taste of a
Right.
food based on the soil, a lot of it is fungal driven. And because the fungi are a big part, I can't go deeply into it. Fungi are deep part of the immune system of plants, which is
Oh,
how a
yeah,
lot of plants
yeah,
get their antioxidants, which is frankly how we get our
yeah.
antioxidants, which are a big part of our immune system.
It's an amazing the
So
analogy between the body and the soil.
it is
You know,
very
the
cool.
biology of it. Let me ask you this too. Dr. Elaine Ingham kind of shared with us many, many, many years ago that in turf, and if you think of a grass plant on the botanical scale, it's fairly at the lower end. It's not a pine tree or a petunia or a grape plant, and the soils tend to be predominantly bacterial driven. And so when we were, back in 20 years ago or 10 years ago, maybe even, we were doing a lot of tests with Elaine's lab, and we would get the fungi to bacterial ratio, and they were always very strong with what we were doing. But we want to promote bacteria in a grass plant. I don't know if does that ring true to you? Does that make any sense?
Elaine Ingham has done amazing work. The Food
She
Soil Lab
has.
really has moved the dial on things. And like I said, microscopy is terrific. I will have to say I'm a little out of my depth.
That's okay. I didn't mean
Um,
to put you on the spot.
but I will say that almost everybody, particularly people who are growing, the closest thing I know to lawn and turf is pastures, right?
Right.
So
Exactly. Very similar.
a lot of regenerative growers are focusing on their pastures. The problem we not the problem we have with the test, but a pasture has huge even meter to meter variation in it, depending on who just went by and pooped on it, right?
Right.
Um, and what's growing. So using our test is accurate in a pasture, but a little confusing because the microbiome differs so much place to place. But um, but pastures also want to be about one to one, fungal to bacteria,
Okay.
again, because bacteria are very little, right? And fungi are very big.
And
So
bacteria come and go very quickly.
and fungi eat bacteria, right?
Yeah.
And fungi build a lot of soil structure, do a lot of good in the world. But when you look at percentage, and when we do percentage, it's sort of carbon based. So, one not one microgram of microbial carbon from a fungi is lots and lots of microbes, right?
Yeah.
Microbial carbon. So it's a pyramid, right? Microbes are much bigger than fungi are there. I will say that in general, poor soil is more bacterially dominant. If turf is bacterially dominant, it's probably because of lack of diversity, right? Which is
It's a monoculture.
yeah, it's a
Yeah.
monoculture. So that's actually your objective. I would say you don't really want to strive to
No,
have
I wasn't trying
bacterially
to suggest
dominant,
that. I was just
but
simply
it's
suggesting it.
probably a
Yeah.
reality, and
Yeah.
you're not gonna get to a 10 to 1 fungal to bacteria, but you might be super excited for close to 1 to 1.
Yeah. Is 10 to 1 where we want to be?
No, 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 no.
Where do we
But
want to be,
I
from a
I
fungal to
do I do
bacterial?
not know, and hopefully someone will
I don't know
comment
that
and let me
anybody
know.
knows the answer to that,
Yeah,
Laura.
I don't
I've
know.
asked that many times.
What I do know is because we have what I like to call heavy hobby cannabis users. They're
Yeah.
probably also heavy cannabis users.
Yes. In many
Um,
ways, they're heavy
and they
users.
grow in real soil in very controlled environments, and they treat their soil like they're the prized chihuahua, right? It gets a
Right.
mycorrhizal fungi treatment every day, it gets tea, it gets all this stuff, and they can have shockingly high microbial contents in their soil and amazing fungal content. But that's not, it's real soil and a natural cycle, but that's not what happens, out
Yeah.
in the field.
Yeah. You know,
We call
you
them sort of curated soils.
Right. You had said something earlier that I was interested in that, bacterial put out one or two things. I mean, one of the things that we talk about is the diversity of the microbiome in the soil. And you know, our approach to that is we're feeding multiple forms of carbon, not just a linear form. We won't just feed a molasses or just a fish meal. We'll feed all of those things. And my understanding, and again, you can correct me if you think I'm wrong here, is that having that level of diversity of foodstuffs is going to produce a diversity of this microbiome. Is that fair?
Yeah, that is absolutely fair. And the other thing to think about is, and again, this goes maybe beyond what we want to talk about is is quorum sensing. So quorum sensing is one of these things that microbes do, and it just means microbes, for the most part, are a self-sustaining population, right? But there are even sort of good microbes can be virulent at times. A good example is strep, the strep bacteria on
Hmm.
humans. We have strep on us right now, right? But we're not sick. But if something happens and the strep bacteria are allowed to reproduce to a certain size
Yeah.
community, then what they do, it's kind of cool, is they, call up all their friends and say, now's our chance.
Let's go. Let's
Let's
go. Yeah.
let's go. And they get infectious, right? They
Yeah.
break through. So you don't get strep through because one strep bacteria landed on you, right? You get it because the ones that you have were somehow allowed or able to get big enough to become bad actors. Um, so if you're only feeding one thing to your microbes, you might be feeding the best thing that a certain microbe wants
Oh,
that
interesting.
could become
Yeah.
virulent. So,
Yeah.
and when we talk about diversity, well, you can't have diversity of crop, you can have diversity of microbes. And of course, I mean, it doesn't take a whole lot of science to think you want to feed it different things. And you also, we don't quite know all the life cycles of a microbe and what they might need at different times.
Yeah.
Um, but yeah, I mean, you do want to feed them different things and not just molasses because that's candy.
That's short chain. Yeah. You know, that might be one of the best descriptions of that I've ever heard. I'm very grateful. I'm going to steal that from you as well. But yeah, I
I
mean,
stole it from my mother, Judy.
well, there you go. And I've stole everything from Jerry Brunetti, but you know, it's always been our belief and our practice as a company to create as much diversity in the food stuff. We can't control the biology, but we can control what we feed the soils where the biology lives. So the more diversity in that carbon food that we provide, I think we're seeing more diversity. Unfortunately, we're not there yet and able to test that diversity, are we? I mean, the biometer is not going to test the diversity of bacteria. It'll just test the volume of bacteria.
Yeah, test the volume. I mean, we do know that the bigger the volume, the more diversity there is, right?
Yeah, that makes sense.
That's just sort of a
Yeah,
mathematical thing. So we're like, look, if you have a very small population, you likely do not have a lot of diversity.
perfect.
And you are likely very susceptible to a virulent bacteria coming in and being able to take over, right?
Yeah.
Which is why low soils get disease and pests more because they don't have that diversity. Bacteria and fungi do self-regulate and they're little armies. They're so cool.
Yes, they are.
But if you don't have enough of them and enough different ones, you really can't. Your little medieval village can't raise a little army.
Yeah, yeah. Can't fight off the
All they
bad
could do
guys.
is give the baker a little knife, right? And that's not the same as having an army.
Yeah.
So absolutely.
Man, your analogies are better than mine. I'm going to be stealing a lot of
I'm
your stuff.
I feel like I'm in a video game right
Yeah,
now.
this
It's
is
very
great.
fun.
I appreciate that. Like I said,
Clash
I'll
of
be stealing.
Clans.
Yeah, as I said to Jerry when he wrote his book, he asked me if he could publish it. And I said, you can't because everybody's going to know I'm a fraud because I've stolen everything that you said. But that's good. We're just trying to share information. What's the most practical way to use the microbiometer? I mean, how often, when, I mean, and again, I'm talking to a sports turf client base, but in your experience, how are people actually using it? How often, frequency? Give me
Yeah.
some ideas as to the practicality of the tool.
So I think sort of just initially, if you're a person that has it, the best thing to, and sort of the most efficient way for you to figure out if it's a tool that will help you, right? It might not be a tool that will help you, depending on what you're doing. The best
I
thing
think
to
it
do
will.
is, I do too. We wouldn't be selling it, but
Right.
I,
I
you
wouldn't
know,
be talking to you if I didn't think that.
yeah. The best thing to do is say there's a turf company that's struggling with adopting a new practice, right? They have a field that is primarily chemically managed or something, and you're talking to them about using more biologics products, right? The nicest thing to do is what I call doing experiments. So you can do an experiment in time or space, right? So I like the ones in space better because time in the natural world has weather in it, which is hard. So if you have one area where you say, look, you know, Roger, let's just try on field, I don't know, I know nothing about golf, on hole 6 to try a different program, or, let's cut back on this and put some more natural fertilizer in. What you do to those two places, one being old school managed and one being the new, is you test on the same day, right? Same weather conditions, apply, and then before treatment, and then you treat them and you test the same spot, right? Like, remember where you went. It's not that hard. And you test it 2 weeks later, a month later, 3 months later, right? To say, look, on the place where we used some biological products to feed the soil microbes, you can see, and I have this terrible like 1970s slide here. I'm so tech savvy. I know. This is a graph of a blueberry plant with and without biochar, which is a terrific product as well. And you can see this is spring, summer, and fall. In the summer, microbes naturally go down. It gets dry and there's drought.
Hot, dry,
and in the fall, they go up. And you can see that the overall numbers for untreated versus treated, right, are lower. So this is your sort of gain in microbial activity. And then really what you want to do is say, as a business, and you spent 20% less on inputs, right? You reduced your toxic runoff, which the community and the golfers do not like, by X. And hopefully this field looks as same or better than the one that was more expensive and more harmful to both the environment and your image as a soil steward, because golf courses are soil stewards. They maintain a huge amount of land in this country.
Big open space, big community.
They support birds. They support a lot of things. And I will also say, I know you say there's no diversity, but a lot of them are going to putting in, areas where there is natural habitation.
No, there's a lot of that going on, but
there's a lot. I mean, it makes it more beautiful. It makes it more interesting. It makes it ecologically more sustainable.
Right.
And I mean, and also less to mow, which is probably their main objective. So what I would say to people is, you're using it to assess your practice because you started to believe in microbes. You want to improve the microbial life in the soil because you think it's going to do other things for you, right? We don't just love microbes and want them to flourish, right? We're doing it for,
we want them there, and we want them working hard every day.
Yeah, we're doing it for real business, financial, and ecological. So say, oh, I can see that these products are working, and I will work to roll them out in other places. Okay.
Yeah.
And the nice thing about doing it in time is that, if there's a drought or some other factor, then both places might have gone down, but you can see the place where you used a biological went down by less, right?
Right. Yeah, Mother Nature has something to say.
Mother Nature is in there, yeah.
But I mean, just to get an understanding, I mean, if I want to build a program for somebody, should I test an area that obviously I would test, a controlled area, the same area, but would I do it once a month? I can't imagine testing once or twice a year is really going to bring me a lot of value.
Yeah, I mean,
I'm not trying to sell your product or overuse it, but I'm just trying to get the data that I want.
It's hard for us because we're not agronomists, and we sell in 107 countries. So it's really, really hard for me to talk. I talk to people in Australia and Indonesia, and I don't know what they're doing there. Yes, if you can, and the tests are $7, so there's no reason why you can't.
Expensive.
Where you can't.
It's a 20-minute test. So, I mean, it's no time consumer, really, in
And
the
the
grand
most
scheme of things.
20 minutes, it's about 2 minutes of work and 20 minutes of it
Sitting
sitting there,
around. Yeah, right.
sitting around.
Good point.
I would do it monthly.
Okay.
And perhaps when you do an amendment or something significant, I might do it every 2 weeks just to kind of see how long
Track
or
it.
what is the one of the things that's cool is that when you till, of course, you'll see it to go down. But if you do it responsibly, it'll go back up, right?
Right, right,
So you can say now part of what you want to know is what's the story going on underground? What
right.
what impacts these microbes for good or for bad? So you might be like, oh, it's been really dry. Let me test it. And of course, irrigation. I mean, golf courses irrigate all the time.
Yes.
What is the impact of irrigation on
Yeah.
microbes, right?
Single biggest input that they have is their irrigation water. And most of the waters that we work with are not great. A lot of them are rich in sodium and bicarbonates, all of which are going to affect the soil biology. So when you're really droughty and your irrigation water is increasing exponentially, the microbiome is going to be falling down. We've used this. Obviously, we've tested with this with our team of agronomists, and they have done the pre and post earthworks product testing, and we see a bloom. We also have been able to go in when a client has said, this fairway six has got this area that's just something's going on, and we can test that, and then go 10 feet down the fairway and test that, and we've seen a difference. So even in very microclimates, the biology can fall off very quickly, especially on a golf course. So, I mean, it's given us that kind of value in order to say, yeah, there's definitely something going on here. And again, as I talked to you earlier, we focus on that three-legged stool: soil chemistry, physics, and biology. And there's very little support for biology, but it's growing exponentially. I mean, in our business 30 years ago, we were heretics. 20 years ago, and a little bit now. Now you can't throw a dead cat without a company talking about something carbon based. So it's here. It's not going away. But unfortunately, they still don't quite understand that the carbon is about feeding microbes. So bringing this tool to the table, I hope will continue to quantify the fact that we're talking about soil biology. But we
Yeah.
hope to be able to use that for exactly those purposes. When there is a weakness in an area, if we can quantify not only our Logan Labs test, our soil chemistry tests, and nutrient load, but if we can quantify even in the slightest way the biology, and like we started to talk about, I'm hoping in 20 years. You guys come out with some brilliant breakthroughs, and we get better and better and better at quantifying and qualifying the soil biome and the number of bugs in there. And I would hope you would tell me that you would think that's going to happen in the next
Absolutely. I mean,
100 years.
it's very early in the process,
Yeah.
and we one of the projects we're working on is we have all these people out here getting soil biology data over time, over place, lots of data. We have over 100,000 data points across the world. Is to say, okay, now that data,
Let's
particularly
use
with the
it.
help of
Yeah.
machine learning and AI, can really be
Wow.
integrated with all kinds of other things. And biology has been sort of the missing piece, right? We have chemical, we have
Yeah, of course.
weather, we have,
And it's hard
all
to
kinds of
quantify, and it's
hard to
esoteric,
find.
you
Yeah,
know?
and different methods do different things. So, while our method has pros and cons, they're
Right.
comparable to each other. So
Yeah.
all that data is on the same
Wow.
scale and can be used. I will tell you one more thing, which I should have said before for turf people. When you're doing your sampling, because you have so much sand,
Yeah.
what you want to do is what kind of the cannabis people do. Sorry to put you in the same
That's
boat.
okay.
Is when you go out
Most
and
of
take
us
your
are.
sample, yeah, pull up pull up some roots. Okay,
Yeah.
microbes live right around the root sphere, and because you have so much sand, it
Well, we
your
also
numbers.
have we also have about an inch of thatch layer, which is very lignous,
Mm,
undigested carbon, root system that hasn't been
yeah,
digested yet. So I wouldn't think there'd be the
yeah.
biological environment is not as strong there as it would be around the
I
roots.
mean, what you might want to do is I know you get in trouble for it. Pull up, a little divot
No, we
of
can do that. We can probe. We
You
can
can do that,
probe.
or take
Yeah.
one that's some
We,
rude. golfer
Yeah,
knocked
we'll
up.
take a sample. That's not hard.
And you do kind of want to scrape the soil. You only need a half a cc for each test. You do really want to, in a way that other people maybe wouldn't, really want to just not pull up a bucket of sand and test it. But
Right,
you
right.
want to say what biology is available to the roots out there.
Which,
And
yeah,
I have to go around the roots and spend a little bit of time, a minute or two, pull up a clump. There's a little spatula in here. You can scrape off some soil and test that,
oh, I
because
like that.
that,
Yeah, that's good. That
yeah,
makes sense.
that, and do it every time, because,
Yeah,
you know,
yeah, that makes
but
sense.
really pay attention to your soil sample, because when you do a chemical test, it doesn't quite matter, right?
Right.
Especially because the way we spray chemicals is sort of uniform. Like in the real world, chemicals are
Yeah. But even when we do a
uh,
standard soil test, I mean, we're testing, to a 6 inch depth, and it's really the mineral content of the soil, where this is a biological content.
is a biological,
But
yeah,
you know,
yeah.
I would love to, and we can talk about this down the road. But I would love to work with you and help that database, because I would love to take what we're doing, and I'm hoping that we can get a lot of folks to go down this road, and we're certainly going to go down this road. But I would love to start really taking our data and putting it into the bigger database and seeing what ultimately this is going to do. I mean, I think intuitively we know that when we feed the soil, biology improves. I mean, we see the results. We see that the turf is stronger, it's healthier, there's less disease, there's more water holding capacity. But having that level of quantification, you know, everybody wants to know that. They want to know that, hey, is this really doing anything? Well, you see that it's doing anything, but that's not always good enough. So I am very excited about all this, Laura, and I'm thrilled that you were able to join us.
Yeah,
And
thank
I
you.
and I want to encourage. I mean, like I said, we're going to have all of our guys will be equipped and hopefully get out there. I'm going to try to get some of my sales reps to do the same thing. The guys that really get there's a percentage of any industry that they just get really excited about this stuff. And
Yeah.
as I talk about it, I'm gonna bring to the table. And I do want to announce that you were gracious enough to, if anybody is interested in purchasing one of these biometers, they're not expensive. They're really easy to use. You can find it at microbiometer.com. And if you put in the code Earthworks20, Laura will give you 20% off on buying a biometer. And I would
Yes.
encourage you to do it. And if you do that, please let us know so that we can gather that data and start to understand that. But I think I might need to have you back someday, because this was great, and it's such a big conversation. And like I said, I hope that in my lifetime, we continue down this road. The work that your mother did was just brilliant, and I'm so thrilled that you're able to continue that work, and I'm sure you are as well.
Yeah, yeah, it's wonderful.
And it's just, and there's just such opportunity and so much excitement in both regenerative agriculture, and a lot of folks like me and my team that really get excited about this, and we want to see this really proliferate, and I don't mean that as a pun. But we want this to continue to grow so we can get things going. Laura, I can't thank you enough for joining me. It was a pleasure to meet you at Acres. I will be in touch. Actually, I'll probably be in touch through purchasing a couple more, because I got a
Oh,
couple
good.
more guys that need them. But thank you so much. And again, if you haven't looked at this, go to the microbiometer.com and check out their website. It's pretty fascinating. And it's like I said, the meter is what, $150?
135, and we
Even
ship
better.
we ship in the US for free. Right
Perfect.
here. We manufactured ourselves too.
Yeah.
So supporting American manufacturing.
There you go. And it's, and the nice thing about this is it really is an easy thing. I mean, you just pay attention to what you're doing, or get one of the guys on the crew that's really excited about this and let them be the guy that does this. But test sites across the property, and then start building some data, and I think you're going to see some really pretty cool things.
Oh, good.
Laura,
Well, thank you so much for having me. I really appreciate it.
it was a real pleasure. Thank you so much.
Okay, bye.